Welcome to the Fetal Pig Dissection Lab Simulator. Let's jump right in. First thing is to pick up the scalpel, and we bring it over to the little green hash, cut it open to pick up the scissors, and we will do so to the first incision and cut. And these are the cuts that the students in this particular biology class are instructed to make, so they have to learn this pattern. Ready to peel state, I like to call it. So let's peel the pig, peel it open. Notice the sound effects that we put in this. I like the sound effects, makes things more interesting. So now it's time to identify the parts. If we click on respiratory, this is the parts that are available in the respiratory system, and you can see on the left hand side all of these parts. So let's go for the diaphragm, which I know happens to be here. Yay! Diaphragm lights up. And let's try to speed through some of the rest of this stuff. And now we're looking for the gallbladder. Anybody know where that's at? It should be underneath the liver, which is one of the parts. So I found the liver. And there's the gallbladder. So hello and welcome to the Human Anatomy and Physiology virtual lab. We try to make it so that the instructions are not necessarily necessary. Not necessarily necessary. Wow, that's pretty good. We try to make this lab very intuitive. Just like in the lab itself, in the physical lab, they would get a bag of bones that is approximated by this list right here. This list shows up in a random order each time, the same way that a student might find a different order of bones inside the bag. So they're going to grab the skull and place it. I'm going to grab what I think is the mandible. That looks good. And I'm going to drag that mandible and drop it in the... No! The foot is not the right bone. So just like in the class, we do get some feedback saying, please don't stick your foot in your mouth. Not virtually, not in the real world. Not a good idea. So this time we'll look for the mandible, which we get correctly, I hope, this time. I let go of it. So I'm going to go ahead and try to put it together. They can choose to move whichever piece they want. I'm going to just go ahead and connect the mandible with the skull. Doesn't seem like it works there. So now that that connects, and you heard my Android phone just go bing in the background, correct with the phone and also correct down below. We are getting nice feedback that they did in fact complete the task correctly. And off to the right, we get an explanation that we call the virtual professor. To the blood smear analysis microscope lab. So let's start with slide 1. And that loads into the microscope. 10 times magnification is move up to the next objective level, which is 40 times magnification. Grabbing that focus ring, we'll go ahead and get that to the point where you can see what you're looking for. So you can see as I'm hovering over these, they start to pop into a different color. So let's say I click on that, and we found it, a red blood cell. Imagine that, there's many of them there, so take your pick, it would have been pretty easy. Gets a little more difficult later on, but get a little pop-up that describes the kind of cell that you found and a little bit of feedback as to why it's important. Grab the slide and move it around. And how about this little guy? Let's click on him. We found the lymphocytes. And how about this one up here? Let's click that. We found the neutrophil. And these contain faint staining granules, which I guess means that when you pass out, you stain yourself. No, just kidding. A little humor, keep it light. We are about to have a quick demonstration of the Beer's Law and Colorimetry Spectrophotometry Science Lab. We are presented with 6 different vials. We're going to open the spectrophotometer lid. So as you can see, I'm bringing this closer and closer to ourselves. And this is back in the day when we were trying to approximate 3D. We're going to go ahead and place it in the spectrophotometer. Right there, and close the lid. So I'm going to click that. As you can see up above in the LED screen, we have setting blank. This is the same behavior of the spectrophotometer in the classroom. So we've calibrated that. Now we're going to go ahead and open the lid, click back on that cuvette to take it out, and put it back. Once we have the spectrophotometer that's been calibrated, we're going to go ahead and take the measurements of the samples. So we're going to grab this first one here, Q5000. We're going to drop that in the same receptacle that we did earlier with the water. So now that it's in there, we're going to close the lid. Welcome to the Star and Galaxy Colors and Measurements Lab. First step asks us to choose a plate from the tools box below. I'm going to go ahead and click on that first one. The O1099 appears. And now we're going to click on the blue region of the plate to zoom to that region of the sky. Step number 2, that's the part we're interested in. So as I hover over that, we get a click to zoom prompt. I click it, and we get the zoomed in portion. And that brings up the transparency that has 14 circles numbered on it, which will help us to label the 14 stars on the image. So I'm going to grab that and drag it until the entire transparency overlays beautifully on those 14 stars. And we get the transparency and the magnifying glass at the same time. Magnifying glass is over top of the transparency, so we can look for the stars that are highlighted by those circles. I will go ahead and just quickly show how we can switch from magnification level of 1 to 2. Recenter that. See how everything underneath the magnifying glass is magnifying, including the transparency and the numbers. Let's switch to 4. And that's a 4 times magnification level. That's about okay for this star. Let's go ahead and take a look at the fact that 0 lines up with the left side of the star and about 4 lines up with the right side. Let's go ahead and compare it relatively to a much smaller star. How about number 14? Barely read that right now. That ruler is very teeny, as is the star. So I'm going to zoom in to 2 times. And close enough. Let's go to 4. And it looks like that fits in relatively just between half a millimeter, not even. Now, this is where the tool might make more sense to rotate, because this is at an angle. So I'm going to go ahead and do that. I'm going to press the right blue arrow on the top of that magnifying glass ever so slightly until we get an angle that makes more sense for us, and then grab the magnifying glass and bring it down, slide it till we can get even more of it outside of here. Looks, this thing fits within 0 on the left and 1 on the right. It's about a 1 millimeter galaxy. 1 millimeter galaxies are very small, and not very many living things can fit in there. But that's not true. We know that's a distance thing. So this does help in a hybrid situation as well. Not as a complete replacement for the lab in the physical classroom, but also to allow more students to use this without necessarily having to compete. So this has been a Star and Galaxies Colors and Measurements Lab, and this is Rene Vogt-Lowell. Thank you for watching this demonstration.